Hello everyone, and welcome to Meet the Presidents. I am Raj Mukhopadhyay, the science writer for the American Society for Biochemistry and Molecular Biology.
And I'm Jeff Hunt. I'm the Society's Public Outreach Coordinator.
So today we're hosting a discussion between two ASBMB presidents. We have Steve McKnight with us, as well as Jeremy Berg. They will talk about the various activities in which ASBMB engages and their visions for steering the society.
All right, so let's start with Jeremy. So Jeremy Berg here is the immediate past president of ASBMB. He served from 2012 to 2014. He began his research career at Johns Hopkins working on zinc finger proteins and was appointed director of the Department of Biophysics and Biophysical Chemistry at the age of 32, which made him one of the youngest chairs in the university's history. In 2003, Jeremy became the director of the National Institute of General Medical Sciences at NIH, and he served there for 8 years. In 2011, Jeremy moved to the University of Pittsburgh, and he's now the university's Associate Senior Vice Chancellor of Science Strategy and Planning, and he holds a faculty position in the Department of Computation and Systems Biology. Say hello, Jeremy.
Hi, Jeff. Greetings, everyone. Thanks for joining us.
All right, and our current president is Steve McKnight. Steve is our current president, whose term started last month and will continue on till the summer of 2016. He's the department chair at the University of Texas Southwestern Medical Center, and his lab works on metabolism using a variety of approaches ranging from bioinformatics to genetics. Steve is a member of the National Academy of Sciences, the Institute of Medicine, as well as the American Academy of Arts and Sciences. He also has experience in industry. In 1991, Steve left academia to go co-found a company in San Francisco called Tularic, which was focused on using regulation of gene expression to treat diseases. He returned to academia and in 1995, but continues to have ties to industry because he co-founded another biotech company. Say hello, Steve.
Hello, everyone. Happy to be here.
Great.
All right, so we're going to have Jeremy start off with some questions for Steve about his ideas for his presidency as in ASBMB, but both Steve and Jeremy really want to hear from all of you who are watching, so feel free to either post questions on our Google Plus page, tweet them at us at ASBMB, or you can email us at asbmb@asbmb.org. So with that, let's get started. So Jeremy, you're up.
All right. Well, Steve, again, thanks for participating in this. You've just gotten started on your term as ASBMB president. What are your goals for your presidency?
Well, as to strict goals, I haven't laid the vision out perfectly, but I've kind of thought of 3 things that I'd like to work on. And first and foremost, I'd like to communicate to the scientific community and policy leadership more broadly the virtues of reductionist biochemical research, the sort of thing that we do as practicing scientists. Additionally, I'd like to see that we increase the membership of our society. And finally, I'd like to see us increase attendance of our annual meeting, especially the inclusion of trainees and younger scientists. So those are kind of the 3 things that top my list, at least at present.
Well, let me follow up. Tell me more about your thoughts about the annual meeting. I know we spent a lot of time together at the last one, and you know I certainly had a great time, as I wrote in one of my columns. But what are your thoughts about ways of improving the meeting, ways of adding more to it?
Well, to me, it's, you know, that annual meeting is kind of the heartbeat of the society, and I'd like to see the annual meeting be an event that would attract broadly our membership and young trainees and just be extremely vibrant. It's been a very good meeting for many, many, many years, but I think we can make it better. And a couple of things, and you and I have talked about this, one of the things that I would like to do is be able to include more plenary lecturers, the sorts of scientists that are doing rock star research that would really be attractants to the sorts of people who are having to decide between this meeting and many other meetings. So that would be one thing. And as I said, I'd really like to see us involve higher proportion, a larger number of young people, so that those scientific sessions are overflowing.
How about interactions with other societies? I mean, one thing which has been, I'm a longtime member of the American Chemical Society, for example, but for biochemistry and molecular biology, for whatever reason, we've fragmented a bit with the Protein Society and the RNA Society and so on. Are there ways that we can interact with other societies that would be at meetings or otherwise that would be profitable?
Oh, I think you're spot on. So, what's happened over the years is the Protein Society was formed, and that took a big hunk of what we're all about. The RNA Society the same way, the Biophysics Society. So those are all terrific organizations that have verve and spunk, and yet that their success is cannibalized us to some measure. So very early on, after I found out that I was elected to be president, I reached out to the leaders of those societies to begin a dialogue as to whether it might in any way be possible to carry out our meetings concurrently. And I know that's very, very hard because all of these organizations plan their meetings years and years and years in advance, but I wanted to at least get that dialogue started, because if we could in some sense bring them back in the fold or join forces with them, I think it would be mutually beneficial for all of us. That may be chaotic in thinking that could come to pass, but it would be kind of a reversal of the fragmentation if we could someday pull it off.
Well, you're right. I mean, I think you're never going to get there unless you start. So having the conversations, I'm sure, is a good first step, and just have to see where it goes. So how have you been involved with ASBMB over the years? I know your first involvement has not been as president, but...
Oh, yeah. I mean, the late and beloved Dan Lane of Hopkins, who was one of my heroes, sponsored me, and back in the day, you needed a nominator. I think that was sometime in the 1980s, and I would attend meetings. I was somewhat passive in terms of my involvement, but then in the 90s, I was elected to be head of the nominating committee, and that was my first interaction in a more significant way with the society, and led me to arm-twisting Susan Taylor to become president. And so I've been involved for perhaps 30 years or thereabouts, and it's it's our society.
So you mentioned Dan as one of your scientific heroes. He was one of mine as well. It's been sad to think about him no longer being with us. He was such a force for good in so many ways. Are there other scientific heroes that you can mention?
Yeah, I could go on and on and on about this, but I picked out four. Some of whom I knew fairly well, and some that I did not know. But tops of the list to me was Fred Sanger. Fred was a modest guy and a scientist. He probably never had more than one or two people working with him, and yet he, changed the field 2 times in proteins and DNA sequencing, and that led to 2 Nobel Prizes. And so the combination of his modesty and the impact of his work, to me, puts him way, way up there. A second was Seymour Benzer, and I did not know Benzer personally, but I had terrific admiration for him. So as a young physicist, graduate student at Purdue, he almost beat Shockley and Bardeen to the semiconductor. And not many people know about that, but he was neck and neck with him, and that arguably is the biggest discovery of the 20th century. He then went very quickly into phage genetics and got so good at phage genetics that he almost cracked the genetic code using phage genetics. But Nirenberg beat him to it. So that, I guess, cracking the genetic code would be maybe the second most important thing to happen in the 20th century. And then he moved into the genetics of behavior and just tore it apart using fruit fly, learning and memory and circadian rhythm. And so Binzer to me was just tremendous. A third was Charlie Yanofsky. And Charlie I did know, have known, and admired tremendously as a 19-year-old kid. He fought at the Battle of the Bulge in Bastogne against the Nazis, and he comes into research and using microbiology and genetics and biochemistry, he just does phenomenal stuff with total humility. His ability to mentor his own trainees was phenomenal, and I was not even one of his trainees, but for some reason he kind of put his wand over me and gave me confidence that I could become a scientist, so he was tremendous. And finally, Don Brown, who was my mentor and the director of the Carnegie Embryology Department, showed me how to be a leader. And Don was, in and of his own research, a terrific scientist, but he had this knack of being able to mentor and build people's confidence in themselves. So, you know, that's four of among many, but those were four people I just thought the world of.
Yeah, I guess reflecting on a couple of things. In some ways, Marshall Nierenberg's success shows you the power of biochemistry as opposed to genetics. Genetics get you most of the way there, but at some point you just have to start with purified molecules, or at least partially purified molecules, and see what comes out of it. But you know, I share your love for Don Brown, as you know. I mean, that's when we first met, I think. But you know, as a chemist who ended up working on this zinc finger protein from Xenopus, where Don was certainly one of the experts, I had all these half-baked and half-brained ideas about the role of zinc in developmental biology, and I'd go over, you know, with Hopkins and the Carnegie being so close, walk over and tell Don my latest idea, and he'd very firmly but gently point out that I was completely wrong and that made absolutely no sense from a developmental biology perspective. And I always walked away from that feeling good rather than bad, which was a real testimony to his skill, because he basically ripped me apart, but did it in such a non-mean way. Now, it wasn't that it was soft. It just wasn't mean-spirited at all. That I still felt good that I'd learned something from it. So you started off at Carnegie, right? So tell us a little bit more about your path through, you know, some, I would say, sort of non-traditional sorts of institutions in many ways.
Yeah, I mean, I was a graduate student at the University of Virginia with the late Oscar Miller, and Oscar did a terrifically famous experiment. He took the germinal vesicle of a frog egg, the nucleus, and dispersed it in distilled water and put it on an electron microscope grid, and he could see right in front of his eyes in the EM active genes, these Christmas trees that showed exactly what a gene looked like. So having learned or thought about genes the whole time I was a graduate student, I came to realize that the one and only place that I wanted to go was to the Carnegie in Baltimore, because at the time Don and others were purifying genes and studying genes not in the electron microscope, but studying genes biochemically, purifying ribosomal genes by cesium chloride gradients and mapping out their anatomy, just using restriction enzymes collaboratively with Ham Smith and others. And I said, that's the place I've got to go, and I've got to learn how to use biochemistry and molecular biology. And so I went there, and Don let me be an independent fellow. He mentored me, so he deserves all the credit, but he gave me independence. And so I started studying genes. I happened to study the thymidine kinase gene of herpes simplex virus, and you know, you learn molecular biology and mapped out its promoter, and then evolved studies such that I could purify the transcription factors that would drive expression of the gene. And so that first 10 or 12 years of my career at the Carnegie were just fantastic. I mean, thrilling, wonderful. And so I had success as a young scientist, and around the age of 40, had the opportunity to co-found a biotechnology company in San Francisco with Dave Goodell, who was the first employee at Genentech and the person who cloned insulin and cloned growth hormone and literally single-handedly made Genentech. Well, Dave and I got together with Bob Tejan, who's now the president of the Howard Hughes Medical Institute, and we decided, let's start a company. Let's build the company around our understanding of how genes switch on and off. And so I co-founded the company, but lo and behold, I decided if I'm going to join this company, I might as well, pack my bags and move my family out to San Francisco and dive in head first. So I went out there, and I was the first employee, and with the help of many, many people, we built the company. And I did that for 4 or 5 years, and the experience was tremendous in many ways. The for-profit world is different. It's teamwork rather than individualism. It's goal-oriented. And then more than anything, it was kind of the first time I was ever exposed to the mix of synthetic organic chemists and biologists. So I'd always been a biologist, and I hardly knew a chemist. I could maybe pronounce Diels-Alder reaction, but I didn't know what it meant. So for the first time, I saw kind of the sparks that could fly when you mixed really, really good chemists with biologists, and that recognition kind of was transduced when I moved back to academia, and that's here at UT Southwestern Medical Center. And having been tapped to be the chairman of the Department of Biochemistry here at UT Southwestern, it was our vision to build a department that would be composed of equal strengths in chemistry and biology. In other words, put the chemistry back into biochemistry. And many other places, biochemistry departments have evolved away from chemistry, and so we kind of went retro. And because of my exposure to chemists at Tulare, I saw this is really what I want to do, and so we've kind of rebuilt a biochemistry department in kind of the image of the 60s and 70s. It's a real biochemistry department. It's got chemists and biochemists and people who are not afraid of metabolism. And that experience out in California, meeting chemists and understanding how impactful they could be, ended up really directing or transforming my vision for what this department should be at UT Southwestern. And it was a long haul because of the fact that we don't have a chemistry department. We're a medical center. So to try and convince terrific young chemists to come here rather than to go to the chemistry department at Berkeley or Champaign-Urbana or MIT was non-trivial. But over time, we were able to do it. And now we have 80 or 90 chemists working side by side with an equal number of biologists. And so I think we've created something interesting and unique and different. And that's kind of a product of my history.
Yeah, I remember Arthur Kornberg wrote a paper on chemistry and biology, the two cultures in JBC, that highlighted the tremendous potential synergies, but also the cultural differences. I grew up in chemistry departments, and it is a very different way of thinking. And when you can bring people together, it's very powerful. But the cultural differences are pretty, take some time to get over. And back when I was a department chair and trying to build interfaces with the chemistry department, with a real chemistry department, one frustrating interaction that I saw all too frequently was that the reward system in chemistry departments with regard to synthesis was mostly around how complicated the molecule was and how hard the synthesis was and how many new methods you had to develop in order to make it, which was perfectly fine. But when you tried to build an interface with biology, a biologist would say, I need this molecule, or I need a molecule with these sorts of properties. Can you help me? and the chemist would look at it and said, oh yeah, we could put that together in a few weeks, therefore we won't. If it would take us 3 years, then we'd be interested, because then it would be really hard, and then the biologist says, if I'm still working on this in 3 years, I'm not going to be very happy. So, you know, I think I've been impressed with what you've built at Southwestern, because I think you've gotten chemists who really buy into the biological problem as the challenge, but then use their chemical skills in very creative ways to help move the problem forward. I mean, it's a real synergy between the two.
So Jeremy and Steve, we actually got a question from the audience, and it goes back to your discussion about meetings, the annual meeting. We have somebody asking about young scientists who they have limited financial resources, and so the question is, how can you help folks attend meetings when there's so many meetings to attend, but resources are scarce? Do you have any ideas on how to facilitate that?
I've thought about that very hard. I think that's a really good question, and I've written it down right in front of me. And this may or may not be an idea that could fly within the context of our finances, but that is to allow young trainees to attend the meeting tuition free, no charge, with the proviso that they attend the meeting together with their mentor, who is a paying member, not necessarily even a member of ASBMB, but is paying for his or her fees for the meeting. So I think we ought to do everything possible to enable attendance of young trainees and young scientists. That's the future of our discipline, and we've got to get them involved. It's crucially important.
And there also are travel awards both through ASBMB and through other sources. I mean, they're still competitive and so on, but there are ways of getting funds that will help defray at least part of the cost of attending meetings.
Right, and we got another comment from somebody online suggesting that different societies sponsor satellite sessions at each other's meetings as a way to sort of encourage the kind of collaboration you guys were just talking about between biologists and chemists. What do you think of that idea?
Well, I'll give a stab at it. The first and most difficult challenge is to coordinate the meetings temporally and spatially. And I think if we could pull that off, then how you would integrate the meetings like satellite sessions, I think that's the easy part. I think that could be pulled off quite easily if we can get over the hump of meeting at the same time in the same place with some of these other societies.
I mean, the other important point is, ASBMB is somewhat unusual in that the meeting is part of the experimental biology meeting. So we already meet with a number of other societies, some of which overlap quite a bit. Pharmacologists and nutritionists and pathologists and so on all meet together, and I certainly, as much as time permitted, tried to get over to at little bits and pieces of some of the other meetings that are taking place at the same place and the same time. That adds a different dimension to the experimental biology meeting that's not present in other meetings.
Cool. All right, so back to you, Jeremy. We'll keep the questions coming.
Okay, thanks. So, Steve, what else have you enjoyed about being a department chair, or maybe not enjoyed so much? How have you found that?
Well, okay, it's 18 or 19 years. I moved back here in '95, and within a year or so became chairman of the department. And when I got here, it was a somewhat dispirited group, because what had happened is about the same time I arrived, Eric Olson arrived, and we, as an institution, decided to have Eric direct the Department of Molecular Biology. It was our UT Southwestern's first Department of Molecular Biology, and that took away half of the faculty of this department and left behind the more traditional biochemists, and they were somewhat dispirited. It was not terrible, but morale was low, and so I got with him and we talked and said, well, what do we want to do? And I knew what I wanted to do, but I had to get buy-in from my colleagues, and what I wanted to do is to bring chemistry, the discipline of chemistry, right into the department. And people agreed with that, and we set a vision to do that. And then the other thing I said to the department, I said, look, 10 years from now, we want to be recognized as the best biochemistry department in the nation, #1. And one of the faculty members raised his hand and said, Steve, how about top 10? And I said, no, not top 10, but we want to be the best in the country. And so that's not something that's a snap of the finger, and integrating chemistry literally took a decade. And yet, seeing that come to pass and seeing us be able to pull this off has been tremendously rewarding. It's the chemists now are interwoven so closely, and the reason it's been so hard in other universities is because they're separate departments. There's a department of chemistry in this building, and there's a department of biochemistry and molecular biology in this building, and it only works if you're bumping into each other every day and talking and thinking every day. And it really helps if a young chemist's kids are going to the same school as the young biologists. So it's a social thing. There's lots to it in order to see these things weave together. And so it really took patience. It was not going to be a snap of the finger. And yet that's come to pass. And there's something very, very apropos to what you said earlier. Most of these young chemists came from labs with the precise mentality that you articulated, and that is the chemists so proud of making the biggest, most complicated natural product with chiral centers here and there and new methods, and that's the mentality that they brought with them. And over time, they've bought into the idea, of course, being terrific chemists and doing that sort of thing, but at the same time, if they see the opportunity to deploy their chemistry on a small molecule that has entirely new biological activity, they've not only warmed up to that, but they're all over it now. And so it's now 19 years, and now it's woven in to the degree that the chemists, people try and steal them away from us all the time, but they're not going to leave because they have terrifically intimate interactions with equally good biologists here, and the chemistry departments that are trying to woo them away don't have that. They don't have that element, and so all the money in the world, if they don't have that, our chemists are saying, well, if you don't have that, I'm neutered, and so it's been a lot of fun to take something from the bottom. And over this period of time now, I think we're viewed unquestionably as the strongest basic science department in the whole university, and I think that should be the case for a biochemistry department. Classically, UCSF are really good places. It's always the biochemistry department. Columbia, many places, is the biochemistry department is the centerpiece of basic sciences, and we've gotten it back to that point here, and I'm very proud of that.
How have you found, more generally, sort of mentoring young faculty members, and particularly in these more challenging fiscal climate? Is that something that you've gotten pleasure from, or found frustrating, or both?
Oh, I mean, the mentoring thing is the whole job. I mean, you do your best to try and recruit a person, and if you succeed in recruiting them, that first year, 2, 3, or 4, that's the time that you talk with them. You go to their office, or they come to your office. You just really talk and think and help them, get their momentum up and develop their own project. And that's the most satisfying part of the whole job. We kind of have a style here that I think is a kind of a sustainable style, and that is we have really good core laboratories. So we have core labs in high throughput screening, proteomics, next generation sequencing, transgenics. We even have a technology core in X-ray crystallography. So we have these very, very good cores that allows the labs to be fairly small in size. So if you have about 4, 5, 6 people in your lab and access to these technology cores, you can really turn the crank. So, we've got a system where a person here can do pretty well with one NIH grant, maybe one Welsh grant, maybe a partial grant with CPRIT or the American Heart Association. So the demand, the pressure for how much money a faculty member needs to raise is mitigated. And so it's all part of finding a strategy for better of any words, survival, being able to stay alive when times are tough, and having access to these technologies rather than individual labs having to carry each of these capabilities. It's very inefficient, it's very expensive. So if you can have a lean team that's quite good, that has access to these capabilities, you have a much easier time raising adequate amounts of money to stay alive.
Mm-hmm.
So Steve, this is a question that's come up from a few of the audience members. How do you translate what you've been talking about to a role for ASBMB? How do you make those collaborations between the different disciplines? How do you encourage, and get those funding levels back up? What role is ASBMB going to play in this process?
Well, there are kind of two things I'd like to say on that. One thing is, it would be my dream, and I don't know whether I have the strength and willpower to do this, but I'd like to kind of do this at the ASBMB level to bring chemistry back into the fold. I mean, it's thinking and asking for a lot, but let's say I have 9 or 10 chemists that are in my department. I should be lobbying them to be attending ASBMB meetings, to bring chemistry back in the ASBMB, starting locally with my own people, but thinking about our organization and how could we as an organization open up in a manner that would bring the chemists back in. So that's perhaps wishful thinking. That's kind of the first half of the answer, and let's hold off on the second half of the answer. So that would be the really bold initiative.
Jeremy, do you have anything to add to that?
No, I think scientific meetings are certainly one venue, both the annual meeting and potentially some smaller meetings where bringing groups together that haven't been together before, or at least recently, to understand each other's problems and to try to figure out potential sources of collaboration is one role that this society can play. I certainly find, and I think a number of people share this view, meetings where you actually don't know most of the people are in many ways more productive than meetings where you've seen all the same people at similar meetings over the year, and you could probably give half the talks yourself if you've been paying attention. I think the chemistry biology question is an ongoing issue. Chemistry still has a huge amount to contribute to biochemistry and biology, but it's a question of the sorts of cultural changes that Steve has made taking place. I mean, it's happened in chemistry departments to a large degree. And one thing's that when I was at NIGMS, there was a program that had started right before I got there, and chemistry biology interface training grants that was trying to do the same sorts of things of encouraging chemists to really think about the challenges in biology as opposed to just the challenges in chemistry. So it's, you know, there are different roles for different organizations to play to try to build these interfaces and let the science evolve for the benefit of everybody.
So we have another question that came in through Twitter, and the question is about what words of advice do you have for graduate students currently with the current funding climate, and what would you say to graduate students right now?
I guess that's being asked of me.
I'll follow up, so go ahead.
Okay. There are kind of two words of advice to graduate students. And first and foremost is ask the question, do you thrive on science? Do you just love it such that it doesn't feel like a job? I mean, if the answer to that question is yes, then stick with it. You're gonna succeed. Kind of the second thing is the recognition to a young scientist, a graduate student, or a postdoc, is the only way to succeed is you have to make a discovery. You know, yes, papers and meeting presentations and so on, but really what you have to do to succeed in this business, and it's a competitive, tough business, but to succeed you have to make a discovery. And if you make that discovery, the door's open. And so find the right lab, find the right project, find the right mentor, and you got to take a risk. You got to get in there and make a discovery, because incremental assistance to an ongoing project is not gonna differentiate you from the many other people. So you got to find a way to find that sweet spot and get in there and make a discovery.
Yeah, I guess what I would add is, you know, it's always been, Steve's a good example, and in my own way I am as well, of, you know, my career path is certainly not one that I imagined originally. You know, I expected that I'd be in chemistry departments and sort of stay through, and I've shifted careers a couple of different times. And, you know, if I look over where my students have gone over the years, some of them have gone into academia. You know, it's about a quarter. People have gone into academia, people have gone into research experiences in industry or the private sector, about a quarter doing sort of science-related activities but not really research, and then about a quarter doing other things of various sorts. So I think for a graduate student, you can start thinking about, you know, Steve said, you know, explore your passion for science, but also learn a little bit about what life is like in other sectors. I mean, if you can find alumni from your school or from your lab, or people you meet at meetings who've gone on to biotech or pharma or other sorts of areas, just find out what their life is like day to day, week to week, month to month, year to year, so you have a sense of what different career paths are potentially like. And then, as you know, depending on what opportunities appear or what opportunities seem will fit your life the best, you can pursue them in a more knowledgeable way. You know, it doesn't mean that you have to focus all your time on the sort of career development stuff, but just become more well informed. One challenge, of course, of being in academia is most people in academia know academia really well and haven't necessarily spent a lot of time in other sectors, so you may have to go outside to other networks to get good information.
So along those lines, and this is a question I think that we've all had on our minds, is industry is somewhere where ASBMB can play a big role, but doesn't seem to have played a big role. You know, what can the society do to engage more with those people who either are thinking about going into industry or currently are in industry?
I think that's a very good point. And in thinking about it, here's one concrete idea. if we're going to have plenary talks, have some of the plenary talks be the most exciting research coming out of industry. So, for example, of recent, just the last year or two, protease inhibitors to the hepatitis C virus protease have been perfected to the point where they're curing the disease. And that all rests on the shoulders of the virologists who studied the virus, and the molecular biologists who cloned the virus, and the biochemists and enzymologists who studied the protease itself, and the chemists who developed the therapeutics. And so what a wonderful thing it would be to have the appropriate person from industry who could tell the story of how hepatitis was cured by virtue of chemistry and biochemistry. Well, that would be phenomenal, and it would just be more inclusive. And we've got to start doing that. I mean, I think about what is our objective in biomedical research, and our objective is to find a way towards treatments of disease, of untreatable disease. The patients don't care about Nature papers or Science or Cell papers. The patients want treatments. We can maybe participate in the early phase of discovering pathways for treatment, but the people who end up taking it over the finish line are industrial scientists. And historically, there is always some level of contempt of academic scientists towards industrial science. And I see, having been on both sides, that's not only unjustified, but it's idiotic. And so we need to find ways to bring that back into our society, if not for no other reason, to excite our trainees, to show them, gosh, this leads to things. This happens. It's a long, hard road to get a new treatment, but that's our ultimate goal as biomedical scientists, is to help find ways to treat untreatable diseases. And that should be something that's really part of our meeting.
Yeah, I second everything that Steve said. I think there's been a false dichotomy between academic and industrial biochemistry and molecular biology that's been unhelpful to both. And I think, if we can understand, one of the things that I did as ASBMB President, and I didn't get as far as I would like to, but I think getting people from industry more involved in the society and in all sorts of ways, from meetings to leadership roles and everything else, because we have a lot to gain by understanding their piece of the world better than we do, and we probably have things to offer to them that we're not offering because we don't understand what they really need very well. Again, I've benefited a lot from my own students who've gone on to careers in industry, and they've taught me a lot about what their lives are like, and it's both, it's different. In some ways, I'm envious because they're working on something that's clearly going to make a difference if it works. In some ways, it's tougher because scientists have the luxury of redefining the problem. If you hit a rough patch, you can shift gears and study something slightly different. If you're working on a drug development problem, you either make a drug that works or you don't. It's a very much crisper answer, and that's great if you succeed, but painful when you don't. So I think there's a lot to be gained, and I hope that, and I'm sure Steve will continue to reach out to people in industry and find out what the society can offer them and what they can offer the society to improve the whole field.
So along the line, something that's really important, obviously, both in basic research and in sort of the more development stage is policy and funding levels. So, I know, Jeremy, you've had a lot of involvement with this, and Steve, you have some ideas. So, where do you see the society taking their role in the policy sphere?
Well, I'll start with that, and I really want to pay credence to Jeremy for that beautiful essay he wrote for ASBMB today that brought to bear the fact that we don't have things set up right. And that was followed up a year later by a PNAS paper by Bruce Alberts and Shirley Tillman and Mark Kirshner and Harold Varmus. So I think Jeremy catalyzed a bit of some embers in the fire to begin causing some of the leaders, the top people in the field, to recognize and understand that we're not doing things right. And to me, the way I look at the NIH budget, if it's 29 or 30 billion dollars, it'd be wonderful if it were, increased every year by 10%, and I hope that might come to pass. But I look at it in a bit of a different way, and that is, I think there's a very heavy level of inefficiency in the distribution of that 29 billion, and if we could participate in enhancing the money going to the appropriately qualified people, it would be like getting a pay raise. And so I'm quite keen to help in any way this small campfire that Jeremy's lit. And I think if we bring the appropriate leadership, so one of my close friends, Peter Walter from UCSF, is the president-elect of the American Society for Cell Biology. Peter and I spent a week together this summer, and we talked at length about the fact that he and I should help what Jeremy started. That's the best thing we can do is really get a process going to kind of reevaluate how funds are distributed and to do our utmost to help the NIH to spend their money wisely. And so that to me is something I'm very passionate about.
So I would say, I think, these are certainly very challenging times. I think the key word, my biggest priority as president was to sort of get these discussions going about sustainability, because I think it was clear even before the sequester and other things that the system had, the enterprise had grown larger than the resources that were available to support it, and that we had some structural problems that needed to be thought through carefully. As Steve said, those discussions are now, I think, taking place at pretty high levels, and there's more ongoing discussions about how to turn some of the ideas into more tangible action plans. A big challenge in all of this is that Congress has been, when I went to NIH 11 years ago, I was a little bit surprised that Congress seemed to be sort of barely functional, I would say, certainly with regard to their understanding of science and so on. And I think, things were much better then than they are now. So I think one of the challenges is trying to find the right partners on Capitol Hill. And people get the story of the importance of research and biomedical research, both for health and for economic development and economic competitiveness and everything else. The problem is just the overall congressional discussions are not taking place in the right way. The whole system is just screwed up in various ways. But I think we just need to be prepared to have the most coherent, well thought out, well supported, data driven arguments, and then try to find the right people to, both in Congress, at NIH, and in academia broadly defined, and in industry, to figure out how to evolutionarily or revolutionarily shift things back in a direction that's going to be more well suited for growth once things start to turn around on various fronts.
So one final question is that we've talked about the role of meetings and policy and helping push science forward. Can we talk a little bit about the journals and the role they play in pushing science forward?
Well, Jeremy will be considerably more informed on this than me, but there's no question at all that our journals, JBC, the Journal of Lipids Research, and Molecular and Cellular Proteomics are kind of are not only our financial cash cow, but the JBC is kind of our history. It's just tremendous. I mean, going back, I don't know, is it 100 years? Whatever. Some of the most famous discoveries in the history of science were published in that journal, and it's iconic. So it's absolutely crucial that we care passionately about the quality of our journals, and everything about them. I mean, I say that from a historical perspective, that they are the fabric. The JBC is the fabric of the society, but moreover, our economic vitality is largely dependent upon the JBC. So they are of crucial importance.
Yeah, I mean, I agree with that, and I also think these are interesting times for publishing as well. There's lots of discussions now about replicability of science and the peer review process in publication and so on, and ASBMB through its journals really needs to be at the forefront, leading efforts, thinking creatively about how to make the journals best for members of the society, but also as robust as possible in terms of the quality of the science that's published and how it's published and data accessibility and all sorts of very actually really complicated and thorny issues in some ways. I mean, the principles are clear, but the implementation takes a lot of thought and creativity. I think we've been we've had very strong leadership for the journals over the years, and we just need to continue to for the rest of the society to work with the leadership of the journals to help them evolve in the right directions.
Great. So I don't see any more questions arising, so unless you guys have any final points you'd like to make, I think we're ready to wrap up. So anybody want the last word? I'm seeing heads shaking.
Fun. Thanks.
All right. And Jeff, thanks very much.
Great. Well, thank you everybody for watching, and a big thanks obviously to Steve and Jeremy for participating.
And please check back on asbmb.org to see notifications about future broadcasts like this. And any questions that we couldn't get to, we will try to get back to those questions later on. We will get answers to you, and we weren't able to do it during this broadcast.
Great. All right. Thanks, everyone.
Take care.
All right. Thank you.
Bye.